Okay, so we ended part 3 with you drawing this graph in your notes. Let's take a look at it again. At the end of podcast part 4, I asked you if you could see a relationship between enzyme concentration and reaction rate. What you should have been able to see from that graph are some conclusions we need to get in your notes. First, write that the reaction rate measures the number of times an enzyme and a substrate bind and do their specific jobs. Remember, we call that enzyme specificity. And that there's a direct relationship between enzyme concentration and reaction rate. Now, what this means is that as we keep adding enzyme to any system like a cell, more enzymes collide with their substrates and then bind with them. This results in a higher reaction rate. Does that make sense? If there are more enzymes, it increases the percentage of time those enzymes bind with their substrate. Now we can see that now as we look at this line. We now have more and more and more enzymes now binding with substrate. Now, the question is, why is this line not totally 45 degrees straight line? Why is it not a complete straight line? Why is it this little half parabola we see here? That's the real question. Write in your notes that when this begins to level off right here, when this reaction rate begins to level off right at this point right here, it means that there's little or no substrate left for the enzymes to bind with. No binding, no reaction. No reaction means a drop in the reaction rate, right? So the substrate becomes the limiting factor here because it will eventually run out and the enzymes will have nothing to bind with. In other words, not all enzyme molecules can find a substrate to bind with. Does that make sense? So think about this now. If this was our sucrose example, remember sucrose gets broken down into fructose and glucose. So when sucrose binds with sucrase, that's when that happens. So if we have a bunch of sucrose in there and we add enzyme, all right, and we get this original reaction rate right here, at some point in time, all of the substrate is going to be bound with the enzyme. So if there's no more substrate to bind with, then eventually, what do you think will happen to this line? Yes, it would actually start doing this and dropping off because there's no more substrate. Plenty of enzyme. Remember, enzyme doesn't get used up, but there's not any more substrate because now the sucrose is broken down into fructose and glucose. Hope that makes sense. Okay, so we just looked at how enzyme concentration affects the reaction rate and saw a direct relationship between the concentration of enzyme and the reaction rate. And what that really means is as we increase the amount of enzyme into a system like a cell, we increase the reaction rate up to a certain point. So it's a direct relationship up until right here. Now get this graph in your notes, label it correctly, and then let's talk about it. Now this isn't enzyme concentration anymore, it's substrate concentration. So write in your notes that a direct relationship can also be seen by increasing the concentration of substrate. Think about it. Enzymes bind with substrates, then let go and are ready for a new substrate to bind with. So again, we're going to get this leveling out because remember, substrate gets reacted with. And so the substrate changes, i.e. sucrose into fructose and glucose. But the enzyme doesn't. So we can flood this thing now with substrate, and that reaction rate would go up, which would mean this line would shift this way. But eventually all that substrate would get used up, and the line would look exactly the same. So the shape of the line doesn't change, but it gets shifted this way. Okay, so write in your notes that if we increase the concentration of substrate, we increase the number of times an enzyme has the opportunity to bind with it, and therefore the reaction rate increases. So we see right here a situation now where the yellow circles now would be the sucrose, and these enzymes now would be these red cloud-looking things. And if we flood now the environment with more substrate, and you can see there's extra substrate, these substrates are just waiting now to bind with this enzyme. And once this enzyme does its job and releases now its products, fructose and glucose, then this next substrate has a chance to move in. So that reaction rate again goes up, but remember the line would look the same. It would just shift to the left here. It would shift upward. That indicates an increase in reaction rate. So at this point right here, let's write in your notes that it's at this point that enzymes have their active sites engaged, and that refer to this as enzyme saturation. So at this point right here, all enzymes now would be engaged with substrate. Graphically speaking, this is the point at which there's a maximum rate of reaction. We're going to call this Vmax. Write that in your notes. We're going to call this Vmax, the maximum velocity that the reaction can run at is called Vmax. And we'll talk about that a little bit later in this podcast series. Okay, now on to temperature. Yep, get this graph in your notes and label it correctly. So, what's happening here, asks our friend the penguin. Write in your notes that each enzyme has what's called an optimum temperature in which it likes to work. And it's at this temperature that the greatest number of collisions between enzyme and substrate occur. Remember what happens to molecules when we increase the temperature. They start to move faster. If they move faster, if the molecules are enzyme and substrate, if they move faster, we increase the probability or the possibility that enzyme and substrate get together. So if we increase that possibility, we have the possibility of increasing the reaction rate. And we see that here as we look at this graph. So as we increase the temperature, the reaction rate really climbs. And it looks completely different than our previous one where we had that nice little leveling here. So this increase in temperature makes both the enzyme and the substrate move faster, therefore increasing the possibility that they hook up. Also write that in humans, this optimum temperature is between 35 and 40 degrees Celsius. Remember that the average body temperature is 37 or 98.6. Okay, so what happens when enzymes are exposed to heat beyond this optimum temperature? Well, from chemistry, we know that molecules begin to move much faster, just like we mentioned, when that temperature increases. Write in your notes that this increased level of molecules, I'm talking about beyond the optimum temperature, disrupts the bonds in the actual enzyme, the peptide bonds in the actual enzyme, and also between the enzyme and substrate. Now remember that these are hydrogen and ionic bonds, which are the weakest of the chemical bonds. Now as these peptides bonds that hold the enzyme together are disrupted, it loses its shape. Make sure you get that in your notes, because we call this denaturization. Denaturization. We're denaturing the enzyme or changing its shape. Now remember, the relationship between enzyme and substrate is all about shape. Because remember, the specificity comes between the complementary shape of substrate and enzyme. So if we change that enzyme shape, can you imagine what might happen? What I mean is, from what you already know, if an enzyme loses its shape or is denatured, can you predict how this would affect the reaction rate? Write in your notes that if enzymes lose their shape, they can no longer bind with their substrate. No binding, no reaction. No reaction means a decrease in the reaction rate, right? So we see that now in this graph. So it's at this point in time that we're heating now that enzyme beyond its optimum temperature. And we see that reaction rate now starting to fall. So from this graph you can see how denaturing begins to drop the reaction rate. Now the rate continued to decline because more and more enzymes are being denatured. They're not all denatured at the same time, so you don't get a complete drop off in reaction rate. They're being denatured one at a time. Now, let's look at the difference in the reaction rate if we decrease the temperature below the optimum. Can you predict the result? Write in your notes that the rate would decrease as temperature is decreased, mainly because the molecules are moving slower and the number of collisions between enzyme and substrate decreases. Less collisions, less binding. Less binding, lower reaction rate. Now the big difference here is that cold does not denature an enzyme, while hot absolutely can. Make sure you get that down. Now, early biologists thought life would not tolerate temperatures anywhere near 80 degrees Celsius. However, they kept finding bacteria, so they kept on looking. Eventually, they found something called thermophiles. Write that in your notes. Thermophiles. Thermo, P-H-I-L-E-S, thermophiles. Write in your notes that thermophiles are organisms that can live and reproduce near the temperature of boiling water, 100 degrees Celsius. Thermophiles turned out to be an extraordinarily useful group of bugs. That's because their enzymes work in high temperatures, where chemical reactions occur more quickly. The conclusion is that thermophile-derived enzymes are used to convert millions of pounds, for example, of corn into sugar to sweeten soft drinks. Boy, do we need that, huh? We need more sugar like we need a hole in the head. But now we use those thermophilic bacteria to speed up the reaction, the conversions of million pounds of corn into sugar for sweetened soft drinks. Yay! No. Okay, so enzymes work within a narrow temperature range. Write in your notes that ectotherms, here's the word ectotherms, that ectotherms like snakes, here's Mr. Snake or Mrs. Snake, who's, chowing down on a rat. So ectotherms do not use their metabolism extensively to regulate body temperatures like we do, and that their body temperature is significantly influenced by their environment and the temperature of their environment. So, for example, desert reptiles can experience body temperature fluctuations of 40 degrees. That's a 100 degree Fahrenheit span. So there can be 40 degrees difference between, for example, sunrise and sunset. Now, what mechanism has evolved to allow their metabolic pathways to continue to function across that wide temperature span? Enzymatic activity is highly dependent on temperature. The energy required varies with the temperature as well. Write in your notes that enzymes require a lot of energy to do well in warmer temperatures than those that do not need much energy, and that all enzymes must have enough energy to meet their threshold. All proteins have a different threshold that must be met in order to undergo catalytic activity. And the enzymes of cold-blooded animals have a much lower threshold than do the enzymes of warm-blooded ones. Okay, that's it for part 4. So take a picture of those notes, submit them to Moodle, grab that study guide and answer the part 4 questions. And we'll see you again in part 5. Bye.